everyone, and welcome to our Discover Science webinar series. If we have not met before, my name is Maureen McDonald, and I'm proud to serve as the Dean of the Faculty of Science at McMaster University. I'm wearing my Mac hoodie this evening. We are a community of 9,000 undergraduate and graduate students, 350 faculty and staff, and hundreds of industry, academic, and community partners. Together, we share the same mission of transforming our world through science. Our Discover Science webinar series will introduce you to exceptional researchers who, together with students and partners, are advancing that mission every day with breakthrough discoveries. In this installment, we'll be looking at the amazing advances happening with radiopharmaceuticals in our nuclear science program. A cornerstone of that program is McMaster's nuclear reactor. Our research reactor is a leading supplier of life-saving medical isotopes used in cancer treatments, as well as many other applications that I'm sure you'll be excited to hear about. Last week, the Ontario government announced $6.8 million investment in our reactor. That investment is part of a $25 million project that will allow us to increase both the diversity and the amount of isotopes produced. And earlier this month, we welcomed Indigenous leaders from across Canada to tour our reactor. These leaders are exploring how small nuclear reactors could cheaply and safely power their communities in the future. The message from this meeting was very clear. There will be no path to net zero without nuclear energy, and there is no path to nuclear without Indigenous communities. On that note, I want to acknowledge that McMaster University recognizes that we are located on the traditional territories of the Mississauga and the Haudenosaunee Nations, and within the lands protected by the Dish with One Spoon Wampum Agreement. Thanks again for joining our Discover Science webinar and sharing our passion for breakthrough research and science that transforms our world. I would now like to turn things over to Dr. Karen Stevenson, Director of Research Platforms Support at McMaster. Dr. Stevenson will provide a brief overview of the university's nuclear reactor, and then she'll introduce this evening's speaker, Dr. Saman Sadeghi. Karen?
Great. Thank you, Maureen, and welcome everybody tonight. I'm excited to be here and share a little bit of information about our nuclear facilities. I'm going to share my screen here. I just wanted to highlight a couple of things about our nuclear facilities that we have here at McMaster. So our campus houses a unique suite of world-class nuclear research facilities that really enable discoveries in clean energy, material science, medical isotopes, and radiopharmaceuticals that we're going to hear about tonight, environmental toxicology, and nuclear safety and security. McMaster is a leader in supplying medical isotope I-125, which is one of the top 10 isotopes used in medicine worldwide. Medical isotopes produced out of our reactor on campus treat approximately 70,000 cancer patients each year. And I wanted to highlight a recent announcement that we made as we partnered with Terumo and Atom V Global Radiopharma Inc. to produce 2 medical devices using the medical isotope holmium-166. One thing that's really great about this project is it's a product that comes from Europe, that because we're producing it in McMaster and producing it in Canada, it means that we will be a site for their global clinical trial. So by producing things on campus, we are helping enable and bring clinical trials and new treatment options to Canadians. Another thing I would just want to draw your attention to is at the bottom here. If you're interested at the end of this, you want to hear more and learn more about McMaster's nuclear operations, we do offer tours. And so I just have the little link at the bottom there at the email. You just send that email and you'll be able to find, organize a tour for yourself. At this point, I'm going to hand it over to a video so you can get a better idea of what's inside of our nuclear operations and facilities.
My name is Erica Dow, and I'm a radiation sciences graduate here at McMaster University. McMaster is Canada's most research-intensive university and consistently ranks among the top 100 universities in the world. And it's home to Canada's most powerful nuclear research reactor, where Nobel laureate Bertrand Brockhaus advanced his work on neutron scattering, and where top nuclear experts are using its power to study materials and nuclear safety, solve complex environmental issues, and produce life-saving drugs. Let me show you how they're doing that. McMaster University established itself as a national leader in nuclear science and engineering in the 1950s. Our researcher success has earned them international awards and accolades, and significant investment from the government. We've built a unique and world-class suite of nuclear research facilities to support research in clean energy, material science, medical isotopes and radiopharmaceuticals, environmental toxicology, and nuclear safety and security. McMaster's nuclear research infrastructure provides unique learning experiences for our students. Both undergraduate and graduate students engage in innovative, hands-on, interdisciplinary research projects that complement their in-class studies. Here at MAC, we conduct research that advances society. We collaborate with hospitals, governments, institutions, and businesses to solve complex problems, enable positive change, and advance the health and well-being of our society. McMaster is a trusted and valued research partner, and we've earned our reputation as a Canadian research powerhouse. One of my favorite things to do on campus is to bring visitors inside our nuclear facilities. Our doors are open to elementary and high school students and teachers, aspiring scientists, and community members to see and learn firsthand what we do here. We view community engagement as a natural and essential extension of our educational mandate, as we seek to demystify nuclear science for Canadians, while helping shape policies and practices that are firmly rooted in evidence. With all that's going on here, with new discoveries being made every day, I'm so proud to be a part of a university that is working to create a brighter world.
Great, thank you for that. I hope you enjoyed that. Now we're going to hand it over to our speaker tonight, Dr. Sadeghi, and he's an associate professor in the Department of Chemistry and Chemical Biology here at McMaster University, where he runs the Molecular Imaging and Radiopharmaceutical program. Before he came to McMaster, he was an associate professor in UCLA and also served as the director of the Amanson Biomedical Cyclotron facility. He also maintains a connection still to UCLA as an adjunct professor. So I'm going to hand it over to Sam today, and I'm looking forward to hearing all you have to say.
Thanks for the introduction, Karen. We have also put together a short video. Maybe we could start with that. That sort of shows a bit of the work that we do in the preclinical radiopharmaceutical development space. So maybe we could start with that. The McMaster Radiochemistry and Radiopharmaceuticals group develops molecular imaging agents for in vivo visualization of biological mechanisms of disease and targeted radiotherapeutics to treat cancer. Our team has a strong track record of discovery, development, clinical translation, and commercialization of novel radiopharmaceuticals.
The process starts with our designated cold chemistry or non-radioactive labs. Here we are set up to build or modify compounds that establish the foundation of our radiopharmaceuticals. The next step in our process can only be facilitated by the availability of various radioisotopes.
McMaster University is home to a suite of world-class nuclear research facilities that enable innovation in radiopharmaceuticals. The McMaster nuclear reactor is Canada's most powerful research reactor and the nation's only major neutron source. The McMaster Radiochemistry group also has access to a range of generator-supplied radionuclides, including technetium-99m and gallium-68.
Here in the Nuclear Research Building, we have several CNSC licensed hot labs for handling radioisotopes, where we can add a radioactive label for either imaging or therapy to a targeting molecule. After radiolabeling and characterization, the radioligand is validated for target specificity in vitro assays. Next, we establish a whole body biodistribution profile at different time points by injecting a very low amount of the radiolabeled targeting molecule intravenously. This allows us to determine the distribution and clearance patterns, telling us how the probe circulates throughout the body and if it accumulates in off-target sites. If minimal off-target uptake is observed, we can move on to imaging studies using mouse models of disease and higher dose of radiolabeled compounds.
We use 3 types of in vivo imaging techniques: SPECT imaging, PET imaging, and finally, photoacoustic imaging. In addition to our focus on molecular imaging research, we also work on targeted radiotherapy with alpha emitters and beta emitters. These radioisotopes are directed to the site of disease in the same way that our imaging tracers are. However, the emitted radiation with radioligand therapies can induce cell damage, causing cell death, with the ultimate aim of reducing tumor size and eventual remission. Single photon emission computed tomography, commonly known as SPECT imaging, uses gamma rays to provide 3D images of the distribution of radioactive tracer molecules that have been introduced into a patient or an animal. Positron emission tomography, also known as PET imaging, is like SPECT, but it tracks photons generated from the collision of radioisotope-emitted positrons with electrons in the surrounding tissue. The photons are emitted 180 degrees from each other and can therefore be detected in coincidence. In addition to our PET scanner, we have a Vivo Laser X photoacoustic system. The technique exploits the thermal relaxation of light-excited molecules to produce detectable ultrasound waves.
Our efforts to develop clinically relevant molecular imaging probes and radioligand therapeutics are supported by 1. Access to both a cyclotron and a reactor, which we have on campus. 2. The unique suite of radiochemistry facilities at McMaster, which include licensed labs and modern discovery and characterization tools. And 3, and most importantly, the motivation of our students, research staff, and the strength of our collaborators. Most of our projects benefit from collaborations with other McMaster experts in areas ranging from engineering, chemistry, and cancer research. Previous achievements include the creation of the Center for Probe Development and Commercialization, CPDC, which is a national center of excellence, and Fusion Pharmaceuticals, which is a publicly traded company by Dr. Valiant. Thank you for watching that. I'm going to share some slides now. Well, thank you all for coming today. I am going to talk a little bit more about some basic science advancements in radiopharmaceutical development, highlighting some of our research, and for the discussion and answering questions, I can focus a little bit more on clinical applications. The examples I'm going to show today have not been translated to the clinic, any of them yet, but they are in various stages of preclinical development and clinical translation. As Karen mentioned, I'm very happy and glad to be back in Canada. I got my PhD from University of Toronto, and then I spent about 10 years in Los Angeles at UCLA, but very happy to be back here. What makes us unique at MAC is our ability to do research with a very large range of isotopes. We have access to both a proton source from the cyclotron, as well as neutron sources from the reactor. So it gives us access to a very large range of isotopes, from imaging tracers that we can produce to therapeutic radiopharmaceuticals. And we have facilities that can handle these radioisotopes. That makes us very unique. It's not usual to be able to handle isotopes from short-lived F-18, even carbon-11, and going all the way to lutetium and zirconium and longer-lived isotopes like I-125. The type of research that we do, and I'm going to draw on examples to sort of highlight this type of research, draws on some very basic science. The students that come to our lab, almost none of them have the background to be able to do the research as they begin, because we draw on basic chemistry, synthetic chemistry, health science, biochemistry, cancer research, and even engineering. We'll have 2 students that are joining us from the Department of Engineering, and I'll draw on examples to show how we bring all of these interdisciplinary researchers together. The advantage is that the training program that we offer is also very unique, and it's supported by industry through collaborations with people like Fusion Pharmaceuticals and MITAX grants. And the people that come out of the program are in high demand, and it's an area that we don't have enough highly trained personnel in. So there's a good training program that we offer through our PhD and graduate program. After the basic science and basic chemistry, we do preclinical validation. This is mostly with mouse models of disease to identify whether we can target sites of disease, both for imaging and for therapeutic purposes. And there is a bit of a lack of resources in academia and industry to do this type of preclinical validation work. So we actually offer that kind of research for industry collaborators as well. Which is a nice opportunity for our students to also get involved with industrial applications. This is where industry has a new radiopharmaceutical or an imaging tracer, and they want to validate whether they can target sites of disease specifically. So we can do that type of work for them, as well as do it for our own research and novel radiopharmaceuticals that we develop in our own labs. From there, we go to clinical translations. At MAC, again, one other aspect that makes us very unique is that we actually have good manufacturing practice labs within our building. This is the requirements of sort of very stringent requirements that are needed to produce something that we can actually inject into patients. So it's very different than the type of work that we do in our lab. Requires strict requirements for coming in, going out, quality control, assurance, regulatory, and repeatability, sterility. So we have all of those resources at MAC, and we have been at the forefront of actually producing imaging tracers, PET imaging probes, and supplying it not within Ontario, but outside of Ontario as well. This was through the Center for Probe Development and Commercialization, which Dr. Valiant started. And now that has transitioned into AtomV, so that there has been quite a bit of a history of commercial translation that has come out of MAC. And from that has actually come a couple of very successful spin-offs. Again, Fusion Pharmaceuticals from Dr. Valiant. They're developing alpha-emitting radiopharmaceuticals for therapeutic applications. And these spin-off sort of pay dividend back to MAC in a sense that not only do they raise our profile and provide radiopharmaceuticals for patients to have an impact in our healthcare, but they also come back and support McMaster through training programs and through industry partnerships, which we're working with right now. And these radiopharmaceuticals, of course, are in much demand. So another aspect of this IP generation that either MAC would license to other companies, or we use to spin off new companies that can take advantage and move these to clinical translation and clinical work. So the type of research that we do ranges from anything from small molecule targeting vectors to larger molecules, biomolecules, peptides, and antibodies, and even larger molecules like nanoparticles. We tend to take these targeting vectors. Some of them come from pharma, some of them come from other research collaborators, and we label them with something that we can see. So that label is oftentimes a radioisotope that we use for imaging, or it could be a radioisotope that we use for therapeutic purposes. This is a form of molecular imaging that is different than what you might be used to in anatomical imaging that you would see, for example, with X-ray or computed tomography. We're actually looking at biochemical pathways of disease. We're looking at upregulation of a particular receptor that's a biomarker of disease. And the tags that we put on there are not this type of molecular imaging that we do is not always based on radiopharmaceutical radioisotopes. We can do that with optical tracers and photoacoustic imaging as well. And I'll show one example of that as we go through these talks. So I'm going to walk you through sort of a transition of projects. And within each of those, I want to point out again, the input from basic science, biology, and engineering as well. So this is one example of a collaboration that started with a lung cancer researcher at UCLA, Dr. David Shackelford. And we were seeing essentially that lung cancer, non-small cell lung cancer patients, some of them were FDG negative. FDG is essentially looking at glycolysis, upregulation of glycolysis and metabolic activity, which is a hallmark of cancer. But as we were injecting patients with FDG, this is fluorodeoxyglucose to look at upregulation of glycolysis, there were a subset of patients that were FDG negative. And we knew that these patients had cancer. The tumors were even showing up on CT. So we knew that they had tumor, but they were not active glycolytically. They were not taking sugar as a source of energy. We developed a different hallmark, a different tracer to look at this hallmark of cancer, looking at mitochondrial activity, oxidative phosphorylation. Without going too much into the details, this was a cationic vector that was attracted to the membrane potential that an active mitochondria maintains for producing energy. So it was a tracer that was looking at oxidative phosphorylation. And as we were looking at animals, for example, as we were growing these non-small cell lung cancers in these mice as a preclinical model, for example, in mouse 1 that I'm showing up there, in blue, I'm looking at oxidative phosphorylation with the mitochondrial tracer. You would see that, for example, mouse 1 would have a tumor that is negative for this OXPHOS tracer, and it was positive for FDG, it was positive glycolytically. Yet another mouse that was essentially grown in the same way, was injected with the same type of cell line xenograft, was negative for FDG and was positive for OXPHOS. So we were seeing a heterogeneity, sort of a phenotypes. We were seeing phenotypes of cancer that were either positive for one or the other, like a metabolic tracer. So highlights the importance of having multiple tracers that can tell us about the biochemistry of disease and go beyond just glycolysis. And this type of phenotyping goes even more in more detail. Even within certain types of tumors, we have heterogeneity within tumor cells. So there are regions of tumor that we talk about cancer, and we talk about particular subtypes of cancer. We talk about lung cancer and particular biopsies of cancer, but not all cancerous tissue is the same. There's in fact heterogeneity even within a certain site of cancer. And we saw that in our studies with this mouse model, that even within spatial differentiation and heterogeneity within a tumor, that there were areas that were FDG positive, that were glycolytic, and there were areas that were FDG negative and OXPHOS positive, that depended on mitochondrial activity for generation of energy for these cancer cells to survive and to spread. And that type of tumor heterogeneity is important. I'll come back to it in an effort to guide biopsy through positron emission tomography and molecular imaging. Another example, again, this is going to be somewhat of a science heavy talk as I'm talking about sort of almost the cutting edge research that we do, but at the end I'd be happy to answer more questions about more clinical routine type of imaging and therapeutics that we do as well. This is another example of a small molecule tracer development that is being pushed forward by our postdoc Daniel Hernandez. He came from a very famous lab in Europe, Roger Alberto, doing inorganic and technetium chemistry. So there's a new way of essentially chelating Tec-99m, which is a mainstay of SPECT imaging, and we are trying to label tracer molecules like DOPA. DOPA is another one of those molecules that a while ago, back in the 80s, put MAC on the map. We were one of the first institutes that labeled DOPA with F18 and had clinical images with DOPA. Today it's used for looking at different types of CNS diseases, for example, and we're trying to make it more accessible. Technetium-99 is one of those widely available isotopes that you neither need a reactor nor a cyclotron on sites. A lot of places have it, a lot of clinics even have it in the US. And here in Canada, we get it from a generator that sits sort of on a bench top. So we have access to this isotope Tec-99m that we could use for SPECT imaging, but it's not very easy to add it to a targeting vector. So with this type of chemistry, this type of a sandwich chemistry, we're developing a method to add Technetium-99 to targeting vectors like DOPA. And here I'm showing examples of verification. Once we add this Tec-99m to the targeting vector in the HPLC, this is a separation technique that shows the purified compound. And one of the things that we do that was also shown in the video, beyond imaging, we can inject disease models and take different organs out and count the activity in each organ to see the biodistribution, to get a map of exactly where each isotope goes in a very quantitative manner. So we can see, for example, what ratio of the isotope goes to the heart, how much goes to the lung, liver, tumor, etc. And with it generates the type of graphs that I'm showing in the lower right-hand corner. And with this, we can essentially show the biodistribution where the tracer goes. And what I'm showing here is that this is a non-disease animal. This was a healthy animal that was imaged with this Tec99 labeled DOPA. And what we're seeing here is very rapid clearance. One of the things we want, which is maybe a little bit different from traditional pharmaceuticals, what we want, particularly with imaging tracers, we want fast clearance. We want something that is injected in vivo through, for example, intravenously, and we want it not to circulate for very long, just to minimize the dose that's given to the patient, but also to reduce the background as we image the particular accumulation of that tracer in a tumor and site of disease. So what this is showing is rapid excretion from non-target organs. It's going to the kidney and to the urine, and it's rapidly excreted. So that means that if you have a disease model that has uptake of this tracer, we can pick it up very easily. For those of you who are interested in chemistry, again, I am here in the Department of Chemistry and Chemical Biology, and that is part of my background. A little bit of interesting chemistry. One of the challenges that we have with tracer development for imaging and diagnostic imaging in particular, one of the most common radionuclides that we use is fluorine-18. This is produced in a cyclotron through bombardment, proton bombardment of oxygen-18. One of the challenges with it is that it's got a 2-hour half-life, which is a good thing because when you inject a patient, you don't want to usually inject them with a long-lived isotope. You want them essentially to go to background. You don't want to have activity in their system for a long time. The challenge is it's only got a 2-hour half-life. That means from the time you produce it in the cyclotron, by the time you do your chemistry, you do your quality control, you create your certificate of analysis, you ship it to a patient, and you do your imaging. So that half-life is working to your disadvantage. That means every two hours is basically you have half of what you started with. So the challenge is we're doing some very complex chemistry here, and we want to do that very rapidly. And that in a nutshell is pretty much one of the biggest challenges in doing radiochemistry, in that you want it to be late stage, you want it to be very rapid chemistry for the labeling. And very quickly, again, one of the challenges with F18 is that it comes in the form, in the most useful form, not always, but in the most useful form, it comes in the form of a fluoride, which is an anion. And the most stable type of a fluorine carbon bond sometimes with these aromatic carbons that have a fluorine on it. One example here is a tracer we're trying to develop for looking at inflammation. This is an inhibitor of an enzyme, cyclooxygenase 2. Again, this was working with a very excellent biologist. This was Harvey Hirschman, one of the inventors and the people who discovered this COX-2 enzyme. And this is an inhibitor, a very good inhibitor of that enzyme. It was actually in the clinic for a little while as a pain medication. In the same way, for example, aspirin is an inhibitor of COX-1 and COX-2. This one was a very particular inhibitor of COX-2. COX-2 is a better biomarker of inflammation because COX-1 is always present in the body, whereas COX-2 is specifically upregulated in its expression as a result of inflammatory pathways. In any case, so we were trying to label this COX-2 inhibitor, and the challenge with it was that the most stable place that you could put the fluoride was on this pyrazole electron-rich moiety. So I had a very talented postdoc at the time, Artem Lebedev, who worked on this electrochemical fluorination, where we oxidized this electron-rich moiety, created this very actually, I think I might even be able to get a laser pointer. That would have made things easier. We created these very labile radical cation moieties that was receptive to the fluoride anion. Otherwise, a negative charge of the fluoride and the negative electron density on this pyrazole would make the two negatives not go together. So creating this receptive cation radicals enabled us to create this F18 labeled and very stable COX-2 inhibitor. And here I'm showing some validation studies that we do with cells. We essentially take macrophages and we expose them to LPS as an endotoxin, essentially to create an inflammatory response. And we see that as we increase the inflammatory response, we have more uptake of our tracer. This is one way of doing cell studies to show specificity. Another example of industry, academic, and cancer biology collaboration. This is with Dr. Sheila Singh, a pediatric neurosurgeon here, and Century Pharmaceuticals. And the study is being led by a talented PhD student, Kevin Visatko. This is for targeting cancer stem cells. We're creating immunopet probes for targeting cancer stem cells. These cancer stem cells, essentially, in a nutshell, are responsible. You could treat the problem was that Sheila was looking at, Dr. Singh was looking at these GBM glioblastomas, and no matter what type of treatment you do with them, they tend to always come back. And the culprits are these cancer stem cells. These cancer stem cells survive, and then they cause the relapse and the regrowth of the tumor. So if you had a mechanism of targeting this sort of a progenitor cells that are the cause of recurrence of cancer, if they had some kind of a biomarker that identified them, and we had some pathway to target them, it would be excellent because we could prevent recurrence of cancer. And so Dr. Singh, in collaboration with Century Therapeutics, created CAR T therapies, which is essentially, you can think of them as they were training these T cells to target biomarkers of these cancer stem cells. One of the biomarkers is an antigen, it's a cell surface expressed antigen, it's called CD133. And through working with them, we got access to the antibodies, basically the vectors, the biomolecule vectors that target this particular antigen. This antigen is essentially a flag that identifies cancer stem cells. And then we had an antibody that was very specific to this antigen. So what we did is that we took this particular antibody, and just like we did with everything else, we added a radioisotope label to it for imaging. And later I'll show that we actually use it for therapeutic purposes as well. Kevin, our PhD student, went through multiple iterations of this with different, so not, the thing to keep in mind here is that as you modify this targeting vector, let's say this antibody, through addition of a label, a radioactive label, you can change its properties a little bit. So that's why it's important to make sure that it still maintains its affinity towards its antigen, and that it's stable in vivo, so that the radio label stays on the targeting vector throughout the study. So he went through 3 generations of these labeled antibodies, from full IgGs to fragments that were slightly smaller and had shorter circulation time. So with the first one, it was very good. We were actually very excited. We put zirconium-89, which has a longer half-life, which is compatible with antibodies that take days to circulate and find a target. So in contrast, for example, earlier we talked about F18 and a 2-hour half-life. You can already imagine that, for example, you couldn't put an F18 on this antibody, RW3's antibody, because by the time this antibody circulates for days, and it really does have to circulate for days, and clears from non-target organs and finds its target site of disease, the F18 tag would have decayed, for example. So it's important to have access to multiple isotopes. And again, going back to the fact that we're fortunate that we have that at MAC, right? We produce zirconium-89 from our own cyclotron here now, and we have access, we have facilities that can handle these multiple isotopes. So we label this RW3 antibody with zirconium-89, and we show that essentially non-target uptake is being reduced. It was very high, it's not very good. It still goes, quite a bit of it goes to the liver, and the tumor uptake was somewhat constant. So as Kevin changed some properties and labeling chemical properties of the label, and the way we add this radioisotope to the targeting antibody, he improved this quite a bit, and we're very happy with this. So now we see much reduced liver uptake, and we see that with time, we actually get more and more accumulation in the tumor. This was excellent biodistribution study. This is the kind of stuff we do with a mouse model, where we take essentially a cell line that we know has expression of this biomarker, the cancer stem cell biomarker, CD133. And we did this in an animal model where we grew the tumor, and then we injected the tracer, and we can see that, yes, the liver, as we suspected, still lights up, but we see quite a bit of accumulation in the tumor. Like Kevin, being a perfectionist that he wasn't quite happy with that. So we went to smaller fragments of this RW3 that we work with, and we managed to further reduce liver uptake while increasing tumor uptake. Here's an example of the 3 generations of the tracers that we developed for imaging cancer stem cells with this approach. And the idea here is, in the purple is I'm showing the tumor here. The idea here is that you want to have as rapid an uptake in the tumor as possible, and as rapid a decrease in the non-target areas as possible. So you want this gap to be as big as possible as fast as possible. And so we achieved that essentially with this generation 3, where we had quite a bit of distinction between the tumor and everywhere else in the body. So you can imagine if this was a patient and we had identified that they had cancer stem cell present, we could target that for therapeutic approaches. I won't go into this too much. We also do specificity studies, for example, where we block the target uptake site with things that have affinity for our antigen to show that we are specifically targeting the cancer sites, and it's not just blood diffusion that's taking our tracer to the site of the disease. Beyond that, we do quite a bit of ex vivo work, and this is important again because some of the gold standards that we do currently for identification of disease, for example, in the clinic, we take biopsy samples. And the problem with biopsy samples, one is that they're invasive, and not necessarily always even feasible, depending on where the site of disease or metastatic sites are. You have to localize where the site of disease is, and you might even miss a metastatic site of disease. But also more importantly, when you take a biopsy sample, you're taking a very small core of tissue that's not representative of the entire tumor. If you recall in the beginning slides, I showed an example of heterogeneity in metabolic profile of disease. Here I'm showing heterogeneity in the distribution of these cancer stem cells. So when we take the tumor out, and this is with autoradiography, where we're essentially showing where the site of disease is. If I was to take a biopsy sample from here, I would imagine, and I would, for example, a clinician might characterize this, that this particular disease doesn't have cancer stem cells, so may not benefit from a cancer stem cell targeting therapeutic. But if you were to take a biopsy from this area of the disease, you would make a different assessment and a different therapeutic approach recommendation. So we're working with Dr. Aaron Fentz there through an Ontario Cancer Institute research grant to combine PET imaging with biopsy guidance in an automated fashion, so a clinician can actually go and take core samples that are guided by molecular imaging, as opposed to, for example, just X-ray or ultrasound. So this type of it's a type of a molecularly guided biopsy sample taking. So there's a term that you might have heard recently beyond all the examples I've shown so far are imaging, but there's also a term that sort of has brought quite a bit of revival to our field. This is the approach in theranostics and precision medicine. And one example I want to show with this is that same antibody that we got from Dr. Singh for targeting cancer stem cells. I showed that we could image sites of disease using that with zirconium-89, which is a PET isotope, and we can construct the molecule in the same way, very similar chemistry, maybe very slightly different chelator. This is a molecule that traps the radioisotope, radiometal, radionuclides. And we could, instead of putting a zirconium in here, which was an imaging isotope, which emits a positron for getting diagnostic imaging, we can now put a damaging isotope. We could put lutetium, which is a beta emitter. And here I'm just showing that, post-modification, this antibody, this is the type of work that we do. We're just showing that essentially its immunoreactive fraction is not changing, that it's interacting with the antigen post-modification in the same way as the unlabeled antibody would. And this is what we want. We want to make sure that we're not modifying affinity by adding all this stuff to it. And here's an example of what theranostic and precision medicine looks like. And this is quite a bit used now in the clinic, and we're just bringing this into Canada for targeting prostate cancer, neuroendocrine cancer, and we're hoping we could use this for cancer stem cell targeting as well through this approach. So the idea is that, as in the previous slides, I showed that, for example, you have a mouse model, you scan it, and you see, okay, this particular mouse has expression of CD133, so it has cancer stem cells. And so we could then swap the isotope with a therapeutic isotope. These are different mice, that's why the tumors are on different sites, by the way. But we could swap the isotope from zirconium to lutetium-177. Again, we're super fortunate because lutetium is now produced from our reactor. So we have capability to produce zirconium from our cyclotron through proton bombardment. We can produce lutetium from neutron bombardment from our reactor. We have facilities that can handle these isotopes. So we're unique in that we're one of the few places that could do these types of studies. Now we see this particular mouse would benefit from that type of a targeted therapeutic with lutetium. We inject lutetium, And here's a sort of a survival study where we see that as we increase the dose of lutetium, and these two mice that have tumor, they survive longer and longer, and that their tumor grows slower and slower. We haven't reached complete remission, but we have shown increased survival and decreased rate of growth of tumor through this type of a targeted theranostic approach. Again, for those of you interested, this is just quite a bit of biology. We are sort of doing some very exciting stuff in taking these tissue samples out and not only seeing whether the expression of particular biomarker is there or not, but also learning a little bit more through these type of a multiplexing immunohistochemistry. We're learning to see what our therapeutic approaches are doing, not only to the tumor, but to the tumor microenvironment through creating hypoxic conditions and DNA damage markers. So we're getting a wealth of information post-therapeutic approaches to see what we're doing to the tumor and how we can better target it, perhaps through combination therapies. Maybe for the sake of time, I know I'm running a little bit late, so this is a collaboration with Dr. Fenstock, and we're looking at Alzheimer's disease. The idea is that through Alzheimer's disease, you have degenerations of these basal forebrain neurons. And one of the hallmarks of these neurons is an expression of a receptor called TrkA. Just, you know, it's the same story. The theme kind of keeps coming back, that you have a hallmark of disease. Again, this comes from our biology collaborator or clinicians, and they know that there's a loss of this particular receptor with Alzheimer's disease. So what is a good targeting vector for this receptor? One of the proteins that is a good targeting for this neuron growth factor, NGF. So again, with the same story, where this is a project by a PhD student, Rojin, and she's taking NGF and adding a label to it, again, for those same types of imaging approaches that we talked about, and maybe potentially therapeutic approaches down the line. How are we doing for time, guys? Do we have time to get in a little bit more science, or should I wrap it up?
I think you have another maybe 5 minutes, Sam, if that's okay.
Okay, so maybe I do want to introduce this. This is a very exciting type of a approach that was not pioneered, but pushed forward quite a bit by Dr. John Valiant. You might have heard his name quite a bit before I came to Mac. He was the one who established and pushed forward the radiopharmaceutical program at Mac. Not only the research component, but also the clinical translation and production and creation of centers like the CPDC. So the idea here is you take that antibody. If you guys remember, the challenge with that antibody, they're great at targeting particular biomarkers and particular antigens. The problem with them is that they, as you remember, they circulate for a very long time. And although that's great, sometimes you don't want to have an isotope on there circulating for days until it finds the site of tumor. So the idea here is that we inject the antibody with 1/2 of a Velcro, and that goes, and we can wait for days. This is non-radioactive. It goes, it gets excreted from non-target organs, it gets accumulated inside a disease, but yet it has this Velcro on it. It doesn't have a radioactive tag, but it has a Velcro. And the other half of the Velcro, we attach a radioisotope to it. And that radioisotope, again, could be an imaging isotope or a therapeutic isotope. And this now is a small molecule. So this small molecule, the smaller you can imagine, the smaller the molecule, the faster its excretion, and the shorter its biological half-life. So now we have the ability to label this with even something like aluminum fluoride, and that's something work that another one of our PhD students is doing, Pamela, that you saw her in the video. And so we can label this with a short-lived isotope. So this gets circulated very rapidly, finds the antibody at the site of disease, and attaches to it right away. So this is a type of a bioorthogonal reaction that we're taking advantage of to enable this type of a platform for targeting disease. Again, the idea is that we have this antibody that we inject with 1/2 of the Velcro. This is a transcyclooctene here, gets circulated in mouse for a long time, a couple of days, and then a couple of days later, in this case, 2 days later, 48 hours later, we inject the radioisotope, and then we target that site of disease with a short-lived isotope. So now so this, you know, before in a couple of slides ago, I mentioned that it's impossible to add a short-lived isotope like F18 because it has a 2-hour half-life with an antibody that has a biological half-life of days. But this type of innovative approach is something that hasn't been in the clinic yet, but we're hoping to see that in the near future. This enables imaging antibodies with shorter-lived isotopes like aluminum fluoride that have much better PET characteristics, more widely available, and have shorter half-lives. As I mentioned, this need not always, molecular targeting need not always be radioisotope based. One of the approaches we're using is photoacoustic imaging. The idea with photoacoustic imaging is that there are molecules that absorb light, but because emission, for example, fluorescence imaging, of in the same way we use PET imaging to find sites of disease, you could use light for identification of sites of disease. The challenge with light is that it doesn't have much of a tissue penetration. So it's very difficult to see light that, for example, fluorescent light that's coming from a targeting vector. However, with this photoacoustic imaging approach, we excite a molecule with light with a near infrared laser. But what we actually observe is acoustic waves, vibrational waves, acoustic waves that come out of that. And acoustic imaging is in the clinic already. It's widely available. So we're trying to work with industry collaborators to move this type of technology into the clinic. It's a non-radioactive method of doing molecular imaging, for example. Here in this example, I'm showing again that sort of a bioorthogonal type of reaction. The targeting vector here is a bisphosphonate that's attracted to bone. So if you have sites of rapid bone turnover and disease, you can target that. Need not always be a bone targeting agent. It could be anything else. And then we add here on the other end our photoacoustic dye. You can imagine if you swap this photoacoustic dye for a PET isotope, we could do the exact same study, but now we could do PET imaging with it. If you swap it for lutetium-177, we could do radiopharmaceutical therapeutics with it. So this study is being driven by another one of our senior grad students, Rowan Swan, and she's made excellent progress with this. Here's an example of a pre-targeting study with this, where we inject that bone targeting vector in a pre-targeting fashion, in that bioorthogonal fashion that I mentioned. It has 1/2 of the Velcro. We inject that. We inject the bone targeting vector, and then a couple of hours later, we come in with the dye and we add the dye, and we can see that it's targeted towards the site of bone turnover. So here again, without going into the details of the graphs, I'm showing that we have specificity for the bone with this type of a non-radioactive molecular imaging agent. We could do multimodality as well, where we could take, this is a particular example of a dye. This is the bacterial imaging platform that we're developing with Dr. Boros, a leading infection researchers at McMaster. Again, we're lucky that we have all of these collaborators, right? So we have these excellent collaborators that have access to models of disease and bacterial infection. And one of the bacterial infection targets that we have is DPA, zinc DPA. This cationic moiety here is attracted to the anionic membrane of bacteria. And again, we think you can take this moiety and we can conjugate it to a photoacoustic imaging dye. And we could at the same time have Tech99M. So we could do both SPECT imaging as well as photoacoustic imaging to get molecular imaging of infection. We're using this technique to look at transplant infection, for rapid transplant infection, to identify sites of infection and intervene quickly post-surgical procedures to prevent further spread of infection. I won't go into this, but again, we also have collaborations with engineers, including ones at McMaster at UCLA. But part of the challenge, as I mentioned, with radiochemistry in a nutshell, again, is that you need to do all of that chemistry really rapidly. When you think about it, it is very complex. We have, a nuclear reaction happening in a reactor. We have or a cyclotron bombardment. So we have our isotope production. We take it into the lab. We have to do some chemistry. We have to add that isotope to a targeting vector. Then we have to do quality assurance. Then we have to package it. Then we have to send it to patients. Then we have to do imaging. So all of that work, really the challenge in doing the type of radiochemistry that we do is to at least make the chemistry as robust and repeatable as possible so that it can be done in a matter of minutes. You don't have time to do a 20-step synthesis. Another part of the challenge is that the chemistry has to be done inside this heavy lead shield. Whereas normal chemistry, you can be in a fume hood and you could add pipette things, add things in and out. With radiochemistry, it has to be automated. The operator doesn't have the luxury of touching things because they need to be safe from radiation. So that chemistry needs to be happening inside of a cave. And so we developed these automated chemistry modules where you just put all the reagents in and press a button and it spits out the final product. The idea was it was almost like an espresso maker where you put the capsule in, and it's not unlike that actually, where the reagents come in a capsule disposable sort of a setting. You plug it in, you press a button, and out comes your radiopharmaceutical, which is now quickly shipped to patients for either imaging or therapeutic approaches. a little bit about clinical translation. That's the last slide I'm sharing, is that it is a bit of a challenge, but there's also quite a bit of opportunities in clinical translation of radiopharmaceuticals. At UCLA, this is now almost 10 years old. At the time, our chair, Mike Phelps, mandated us to translate this tracer to the clinic with a budget of $100,000. Of course, we ate some of the overhead, but we managed to do that. And so this preclinical discovery stage, which is much of the work that I showed you here, can very easily and not without too much expense be translated to first-in-human clinical trials. And again, at MAC, we're fortunate that we have all the resources to be able to do that. We have GMP manufacturing. And so once you validate all of that your targeting vector is going to the site of disease, it's specific, and once you show that you can make it in a feasible way, your chemistry is robust, it works every time. You don't want patients to go to the clinic and wait there, and then you don't want your chemistry to fail on the day of imaging or day of therapy. The challenge is these are sort of freshly baked pharmaceuticals that you're shipping to the hospital. You don't have the luxury of making it 2 days before. You have to make it on the day of. So you need your chemistry to be robust. So once you've done all of that work, then you submit some regulatory paperwork to Health Canada or in the US to FDA, and then you go through the phase 1, 2, 3 clinical trials, and we get to approved drugs. There weren't too many of them. For the longest time, it was just FDG, sodium fluoride, and a number of other tracers. But now these the scope of these radiopharmaceuticals is rapidly expanding to include these theranostic agents that we use for imaging and for therapeutic purposes. Acknowledgement, I have to say, this is limited acknowledgement to the slides that I presented today. But again, much of the work that we do, almost every one of our students benefits from collaboration with researchers in cancer biology, clinicians, and engineers. This is some of our collaborators that helped in the projects that I mentioned in these slides. And this is almost all of our current group. We're missing a few people, 2 or 3 people. And that's John here. He wasn't able to make it on that picture day. Anyway, with that, thank you, and we'll be happy to move to questions.
Great. Thank you so much, Sam. That was really interesting. I learned a few things myself that I didn't know you were up to, so that's exciting. I have a few questions that we've had come in, and maybe I'll start with this one, which I think you kind of addressed, but it's always a good chance to kind of cover the basics. Is how do you find the active molecules to deliver the radioactivity, and how can you tell if the radioactivity only goes to the target?
That's an excellent question, and I think some of the most successful targeting vectors have come from clinicians and from a position of a sort of an unmet need. You know, and the targeting vector ranges from anything. It could be a metabolic tracer, as I showed. For example, it could be glycolytic, oxidative phosphorylation. It could be a targeting vector that targets hallmarks of cancer and surface antigens that are specific to disease. So, you know, sometimes they come from clinicians. We know, for example, that cancer depends on glucose. And so we know that, you know, radiolabeling glucose through just upregulation of glycolytic activity and the Warburg effect, we can see disease with a radiolabeled glucose. Sometimes they come from pharma. Some of these receptors, inhibitors of enzyme activity, for example, are specific to enzymatic expression that's, you know, specific to disease. So pharma, for example, has drugs that are inhibitors of enzymatic activity. They could be both, you know, for example, or receptor binding agents, agonists and antagonists. And we take these and we radiolabel them. We have a very active collaboration now with National Research Council. They have an excellent antibody engineering team that they're developing these antibodies for targeting disease antigens. So now we're collaborating with them. Like, you know, we're asking them, can you give us some of those antibodies? And then we turn them into radiopharmaceuticals. There are some nuances as to what makes a good pharmaceutical as opposed to a good radiopharmaceutical, but there's a lot of overlap there as well.
Fantastic. One of the other questions that came in, which I think is a great one, something that you always have to answer, is how much more effective is this approach than current cancer treatments? And, you know, how could you kind of benchmark that and maybe help add some context there?
That is an excellent question, and one that we often struggle with ourselves. You know, one of the things that actually, believe it or not, held back sort of more widely acceptance of new imaging tracers for PET imaging was that FDG was so successful. It's sort of almost like a catch-all. I showed examples where it isn't, for example, where it doesn't catch disease, but in a large majority of diseases, in different types of diseases, upregulation of FDG can catch cancer and metastatic sites. So the question is what you're developing really that much better? And then that's a good question. Can you justify the cost of potentially millions of dollars for clinical translation that would benefit a subset of patients? So that's sort of the important point to emphasize, is that they do. So this is the idea of precision medicine and personalized medicine, in that, yes, it is more expensive, but a subset of patients, for example, who have overexpression of PSMAs for prostate cancer, for example, like we're talking about here, a subset of patients that have PSMA overexpression, and then we could monitor and provide that personalized precision medicine for them, they do benefit. And now we're seeing that, you know, patient lives are heavily impacted. They benefit quite a bit. They're living longer. They're living a better standard of life, because they're not subjected to potentially toxic chemotherapeutic agents, but it's expensive, right? So it's a bit of a balancing act there.
Yeah, I think there's a bit of sometimes a trade-off between a longer term treatment and a shorter term treatment. And maybe the shorter term is beneficial for some other reasons to our indigenous populations or travel considerations. And so there's a whole combination of things that goes into that decision too, on which treatment option that it is. So another great question here is, does McMaster supply radio tracers to PET centers outside of Hamilton?
That's an excellent question. At the moment, our GMP facility, that's good manufacturing facility, this is a facility that makes radiopharmaceuticals that can actually go into humans, is being operated by a company called Atom V. Now, Atom V benefits from a long history of success at MAC, is an offshoot of CPDC, Center for Probe Development and Commercialization. That center was created by Dr. Valiant, who spearheaded that creation of that center. And CPDC, for example, so right now, that company is manufacturing GMP radiopharmaceuticals. CPDC, when it was created, was instrumental in bringing PET to Canada. At the time, we weren't doing so many PET scans, and part of it was that we didn't have access to these radiopharmaceuticals. So when CPD was created, in large part, provided access to FDG and other PET imaging tracers to all of Ontario. And yes, so we do, and we have a track record and a history of providing radiopharmaceuticals, both for imaging and therapy, not within Ontario, but also globally. Some of these longer-lived isotopes, Atom V is now shipping them all over the world. So they're not restricted just outside of Hamilton, but depending on the feasibility and practicality of shipping it based on the half-life, we do supply the global demand for radiopharmaceuticals.
Yes, I can say on behalf of the reactor, certainly our I-125, we certainly ship it all over the world, from New Zealand, all over Europe, North America, China, all over. So, and then another question that came in, I think this is a great one. What role does medical imaging play? And I'm going to take that as in the broader context of medical imaging, X-ray, MRI, CT, probably versus PET inspect.
So that's an excellent question, right? So it plays a number of different roles. I mean, the idea, for example, if you go for annual screening for breast cancer, right? So that's a type of medical imaging. The difference with much of what I showed here is that while those are more anatomical imaging agents, we're looking at molecular imaging agents that are actually telling you something about the biology of the disease and the biochemical pathways of the disease, things that you can actually target. So we learn, not only do we see that disease is there, but we learn something about the disease in the simplest form. For example, we learn that the disease is dependent on glucose for upregulation and energy creation. So we learn something about the disease, and one of the criteria that, in fact, regulatory agents often assess in clinical trials is, what does it do in therapeutic assessment? So does it change, for example, the therapeutic approach that a clinician would take? And for example, even with FDG, there's a certain percentage of patients that then, after getting a scan, would have a different therapy if they had not had the scan. And so basically they can go on a medical treatment that can benefit them, and they can avoid going on something that's toxic and non-beneficial potentially. And another factor is early detection. So we know one of the things that almost everyone can agree on is that early detection of disease and cancer is going to be that much more beneficial for patients for therapeutic approaches. Another area that we could benefit on is a response to treatment. So let's say you have had a treatment, and have you had, maybe you've had recurrence. We can go back to that example of CD133 for cancer stem cells. So if your disease comes back, why is it coming back? Are we just going to keep hitting it with the same drug? Or do we look for the cause of why it's coming back and whether it is coming back, and then stratifying the treatment strategy for that particular patient? So it definitely plays a huge role. Diagnostic imaging plays a huge role in treatment monitoring.
Yeah, and they work together. Really, the functional and anatomical work well together. You really need both of them. So this is an interesting question. I don't know too much about myself, I'll admit ahead, is how useful is deep learning, DP, for molecular imaging?
That is also an excellent question. So this idea of deep learning is all over the place now, even in synthetic chemistry. You talk to some hardcore chemists, and I was sitting through a lecture the other day, and they straight up, they said it's you can't replace an old synthetic chemist with a computer. but it does play a role, and it's something that we actually have, believe it or not, some funding to do this type of work through Yes, it was through CPDC and Ontario Cancer Institute for Research, Institute for Cancer Research, OICR, where we're trying to find targeting vectors. So it does play a role. We're trying to find basically through targeting vectors through chemical modifications that has the best affinity. And then we model that. Not only do we have the targeting vector, we can model to see, we're going to modify it a little bit, add this arm and that arm, and this is how we're going to radiolabel it. And then we can predict, before doing all that expensive chemistry and preclinical work with a computer simulation, we can predict how good an affinity it's going to have. And that can guide the design of a molecule. I would say I haven't seen any really impactful examples of success yet, but it is an emerging field.
Yeah, I think it is showing up kind of everywhere, but I hadn't yet applied it in my mind to molecular imaging. Last question for the night. How can people get involved in this research, and what can the public do to support it?
That is also an excellent, really good question tonight. So it is part of our mandate to educate and to spread the word about molecular imaging, its benefits, and patient partnership is an important part of our research. In fact, some of our funding agencies require it, and I wholeheartedly welcome it. So I think participating in conferences and symposium like you're doing tonight is the first step, is just to learn a little bit more. And I have to do a little bit better, and researchers like myself have to do a little bit better in communicating our work a little bit more to the public that may not have had years of research experience in this particular area. So and most researchers, I myself, I know I'm open to even if you guys want to reach out to me, either myself or researchers within our lab. We're happy to discuss our research, our cutting edge research, or the things that are maybe even routinely available in the clinic that might be beneficial. And of course, we have outreach programs. We take undergraduate students that, as I said, there's, at least as far as I know, no undergraduate program prepares students to do this type of work. As you saw, there's chemistry, but there's all kinds of radiochemistry. So we take undergraduate students, quite a few of them, that do thesis projects and summer internships. And we're trying to approach it, recruit, even go a little earlier and have some educational seminars for high school students as well. But again, reaching out to researchers is an excellent idea. Participating in these symposiums is an excellent idea. And if there's anything I can do to answer questions, please do feel to reach out to me.
Fantastic. I think we'll maybe end it there. And thank you very much, Sam, for sharing all of the cutting edge research going on in radiopharmaceuticals at McMaster. We're very excited about all the things that are coming in the pipeline, and this field is just growing so rapidly. It's an exciting time to be a part of it. I would like to many thanks for everybody joining us. And if you would like to learn more about Dr. Sadeghi's research, you can visit www.mcmasterradiochemistry.com. And thank you everybody for joining us.
Thanks, everyone. Thanks, Karen.